Hello, and welcome to our Insights lecture series. If this is your first time joining us, we welcome you. If you've been to previous Insights lectures, we welcome you back. As you may or may not know, Sanford Burnham Prebys is a nonprofit medical research institute. That means that we are supported by philanthropy. Without our donors, we can't do the wonderful research that we do. It is because of people like yourselves that make contributions that allows our science to proceed. But that's not the only definition of philanthropy. Each of you watching today is participating in philanthropy just by virtue of your being here and learning more about what we do. You're investing your time in our research, and for that, we are extremely grateful. Beyond that, we ask you to participate in ways that are meaningful to you. Perhaps that is you sharing this video with other interested parties after the event. It might also be making a donation to Sanford Burnham Prebys to support our research. And one of the things you're going to learn about today is that that donation does not have to be for $1 million. Sometimes smaller donations can lead to huge results. Of course, you can also feel free to donate $1 million if you have it. So what do we all think about when we think of February? Valentine's Day. And when we think of Valentine's Day, we think about our hearts, which is probably why February is National Heart Month. Today, we're going to let you hear not only from two of Sanford Burnham Prebys' leading researchers on heart disease, but also from a local physician who is working in collaboration with our scientists, and from a representative of the local American Heart Association about the importance of this research in promoting heart health. So, let's begin. Dr. Evan Muse is currently the lead for cardiovascular genomics at Scripps Research Translational Institute and cardiologist at the Scripps Clinic in La Jolla, California. His mission is to identify risk factors in patients to optimize lifestyle and treatment strategies before the symptoms of heart disease take hold. As a physician scientist, Dr. Muse has helped his team move the needle on patient outcomes using genetics, lifestyle, and health to optimize medical treatment and lifestyle recommendations to individuals. Please welcome Dr. Evan Muse.
Good afternoon. It's great to be here, and thank you for that introduction. You know, I get to start from giving us the 30,000-foot view, if you will, and discuss a little bit about atrial fibrillation as it relates to being a medical condition that many of you may have or know someone with, and then also discuss a little bit about the exciting collaboration that Dr. Colas Larson and I have as well. So usually, I think it's fun to start out with a clinical case. So let's start with this 58-year-old who approached me with a recent stroke. Now, this wasn't in a clinic. It wasn't in a hospital. It happened to be in a backyard barbecue at Encinitas several years ago, right? We're always available, always on call, even at barbecues. So go figure. Anyway, this gentleman, unfortunately, was without health insurance and was an artist working on some creative ventures and just didn't have enough really to make ends meet at the time. He was overweight, inactive, and a former cigarette smoker, but was trying to turn things around. He noted that hospitalization several months ago, he had gone and he felt some weakness on the left side of his body, which many of you all also know is one of the signs of a stroke. He felt sluggish and just didn't feel right, so he decided to get attention at the local emergency room. Their head imaging, which they did, revealed a small stroke, and he underwent 48 hours, which is pretty typical of inpatient monitoring. And he told me, as he told me a little bit about this hospitalization, he said, you know, no one really could figure out why I had this stroke, you know, and I scratched my head a little bit and I said, well, you know, let's see what we can do about that. So as we think of our patient, this 58-year-old, you know, I'll be kind of coming back to this in our discussion to try to think of what other diagnostics or even preventative strategies could we have for this individual. So let's back up a bit and start with what's a normal heartbeat. Okay, the normal heartbeat begins in the sinoatrial node, which is up in the right atrium. That's a electronic pacemaker, if you will, our natural pacemaker that tells our heart to beat anywhere from 60 to 75 beats a minute. Maybe if you're an athlete, much slower than that. It then sends signals to the left and right atrium to beat concordantly, and then passes that signal down to the atrioventricular node, aptly named because it sits between the atria and the ventricles. That signal then gets passed down through the bundle branches to the ventricle to make the bottom part of the heart beat. So you get a coordinated contraction of the top and bottom parts of the heart, the atria and the ventricles, to create the normal heartbeat. And that is represented very nicely, as you can see on the right side of your screen, with the P, QRS, and T wave of an electrocardiogram. Now, atrial fibrillation occurs when we get an abnormal rhythm from the heart rather than occurring at this normal sinoatrial node. Atrial fibrillation is a kind of chaotic, disorganized impulse from the atria, oftentimes in the left around the pulmonary veins, but also can come from the right-hand side as well. This creates, rather than a coordinated contraction of the atria, it's just fibrillating, if you will. And then the bottom part of the heart, the ventricle, is seeing these irregular beats come out, often very fast. You can see the squiggly line at the bottom of that screen representing this fibrillating atria, and then the coordinated irregular contractions of our ventricle there. Now, how do we diagnose atrial fibrillation? Well, we use an electrocardiogram. Many of you may have an electrocardiogram, and thank goodness it was not by sticking your hands and legs into a bucket of water, as it was originally done. Now we have different electrodes we can place across the chest to really detect the electrical signals coming from the heart. And even now we have even things smaller than that, whereas some of you can detect an electrocardiogram using your smartwatch or devices that connect to your smartphone, as you can see here. And that's what I happen to have in the backyard for our 58-year-old with the recent stroke. I placed my device on his hands, and with my smartphone, I read this EKG that you can see here, the single lead ECG. And this very clearly shows an atrial fibrillation slash atrial flutter rhythm with a very irregular ventricular beat. And I told him, I think we've realized why you had that stroke. I was able to get him into a free clinic in North County to undergo the appropriate next steps in testing. Now, it's nice to have new digital devices such as this. Our group at the Scripps Research has been doing a lot of work with small digital devices in health, and even here in clinic as well. I've taken these devices to other areas and sometimes get things that I can't quite make sense of, but this was in fact just a little kitty cat that someone had brought to the office. Don't worry, the kitty cat's doing fine. So who are the folks that have atrial fibrillation, right? Atrial fibrillation is actually very common. It's the most common conduction system abnormality that we see in man, right? In 2010, it was about 5 million individuals in the US, and it's expected to be 12 to 20 million by 2030. Now, we see this in all races and age groups, although we mainly see this in a European population more than other ethnicities. And our lifetime risk, once we get above 90, is somewhere in the 1 out of every 3rd person will have or have had an episode of atrial fibrillation. So you can see this is not uncommon at all. The risk factors that put us at risk for developing atrial fibrillation include things that you probably see your doctors for already. Things like elevated blood pressure, cigarette smoking, diabetes, obesity, and even prior cardiac symptoms such as heart failure or coronary disease. However, the #1 risk factor for this is something that we really don't have control over. Every time we blow out the candles on a birthday cake, we have an increased risk because of age is such a determinant for our risk of atrial fibrillation over time. But we're also learning that there are non-clinical risk factors as well, such as those that are encoded in our DNA. And we've learned that genetics is a really important factor for us to look at when determining what's our risk of developing atrial fibrillation over time. There are now over 142 independent genomic variants associated with atrial fibrillation, and we can use these to develop genetic risk scores, such as what our team did a few years ago, where we did the giraffe study looking at genomic risk markers for atrial fibrillation in a cohort that we did long-term cardiac rhythm monitoring on. And we were able to show that individuals in the highest risk strata, the genomic risk strata for atrial fibrillation, were actually 3 times more likely to develop it during the course of this trial. So we are now learning from genetics and able to take this into the clinic for risk prediction and monitoring methods. But why does AFib matter? right? Just like our patient from the barbecue, AFib puts us at risk of stroke, and that's our number one concern. So when folks have been diagnosed with atrial fibrillation, we want to reduce our risk of stroke as much as possible. It can also cause heart failure and shortness of breath, increase our risk of hospitalizations, and reduce our overall quality of life. So it's definitely something that needs to be treated appropriately with your care team. Now, what are the treatment options available to us in atrial fibrillation? Well, first off, as I mentioned, is stroke prevention, right? So individuals with atrial fibrillation that have enough risk factors for stroke should be considered for anticoagulation, also called blood thinners. And if you've ever watched a PGA golf tournament on a Sunday afternoon, you've seen enough commercials for these medications out there to know what I'm talking about. There's also other mechanical things that can be done, such as using left atrial appendage occlusion devices, but that'll be something you'll want to talk with your physician about closely. Now, the medical therapies for the arrhythmia itself, after we've dealt with the stroke prevention, come in two flavors. The first is either rate control, which we can do by slowing down the conduction through that atrioventricular node to make sure that the ventricle is not beating at 120 or 140 beats a minute. There we can use beta blockers or calcium channel blockers most readily to take care of this. There's also a rhythm control strategy where we can use a host of antiarrhythmics that have been around for a whole long time: propafenone, sotalol, flecainide, amiodarone, and dofetilide, or even take people down a more invasive approach for what's called a catheter ablation. And again, which pathway we choose depends on what are the patient characteristics, what is the structure of the heart on an electrocardiogram, and what are the symptoms that are being created here. But unfortunately, looking at any of these strategies, they've never been married or even determined by one's genetic risk or genetic determinants. And that's something that our collaboration is trying to change. Personally, I like to use our own genetic risk to try to modify our lifestyles, to try to understand our risk, whether that's alcohol consumption, weight, exercise, etc. And we can do this with atrial fibrillation and things like coronary artery disease. But on the bigger picture of things, we are now at a point in science where we can use insights from our DNA, these genomic insights, to help guide us through gene-driven drug design. And I think what you'll hear today from my colleagues, Dr. Colas and Larson, is something getting very near to that, which is pretty exciting. I look forward to spending this afternoon with you and the questions. I'll turn it over to my colleagues now for discussion on some of these things going on in the laboratory, and happy to discuss outside of the conference with you as well.
Thank you, Evan. We appreciate that. We're going to take some questions for Evan, but we ask that you please keep them general. As you can see on the screen, Evan has shared his contact information, and if you have questions about your own medical care or condition, please feel free to reach out to Evan directly or make an appointment to see him in clinic. So, Evan, what I took from what you were saying is it sounds like almost your heart rhythm, I draw a parallel to sort of insulin levels in diabetics, that, you know, in a diabetic, you don't want the insulin levels to get too high or too low. You want to stay in a normal range. And so that's what I was hearing from you with our heart rhythms, correct?
Absolutely, Joe. You know, when we're thinking of that first pathway, that rate control strategy, you know, our goal is to not have your heart rate being, you know, usually over, you know, high 80s or 90s just at rest, right? So if you're sitting in bed watching Netflix or whatever, we don't want your heart to feel like it's running a marathon. That can lead to a cardiomyopathy over time or a reduced function of your heart. So we really like to make sure that if we're choosing the rate control strategy, that we get your heart rate down to something healthy. We have people very often present to the emergency room with heart rates in the 150s, 160s, 170s when they're in atrial fibrillation. So you can see that the safety issues there, wanting to get folks down to a more normal range with a rate control strategy.
Is blood pressure part of that as well, or is that a separate issue? If you have high blood pressure, but your heart rate is within those normal parameters, let's say?
Yeah, so blood pressure is usually thought of as kind of being its own thing, while we want to monitor people's blood pressure as well, make sure that blood pressure is not either too high, which is again puts you at risk of higher likelihood of developing kidney disease, heart disease, and even stroke. But also if your heart rate is sometimes gets up to 180, 160, these very fast rhythms, sometimes there's not enough blood pumping through an efficient heart, and you can actually see blood pressure start to drop. So it's definitely one of the things we pay attention to, and it's related to the heart rate, but usually not one of the first things that we're looking at for our AFib patients.
Got it. We have a question from our audience. What about if the heart rate is low, like 50 was the example given here? Is that good or bad for this?
Yeah, so great question. So a low heart rate can be a good thing, can be a fine, normal thing. It can also be a sign of some conduction system fibrosis. Atrial fibrillation, the cardiac the response in terms of heart rate can be either too slow or it can be too fast. And we see these as what we call a tachycardia bradycardia syndrome for some patients. So everyone could have a different presentation of their atrial fibrillation. I do have enough patients that are in atrial fibrillation without being on rate control agents that their heart rates sit in the 50s, 60s, and 70s without any requiring any medications. So it all depends on one's underlying risk factors and how much conduction system fibrosis that they have. But it's something that we take into close consideration when we're dealing with what our next steps are going to be in our therapeutic pathways.
Thank you. Another question from our audience. What is the difference between tachycardia and AFib?
Yeah, so tachycardia is a blanket term that we use to describe any heart rate that's over 100 beats a minute. We can have tachycardias that are sinus tachycardia, where that sinus node up in the right atrium is telling the heart to beat down the normal conduction pathways, but just at a high heart rate. And you'll see, I'll go into sinus tachycardia when I'm out running on the street or at the gym lifting weights, right? Our conduction system helps us keep up with cardiac output. So there's times when we want to have a faster heart rate to supply the body with blood and oxygen. However, atrial fibrillation can be fast, can be slow. Atrial fibrillation is used to describe the rhythm itself. And on our electrocardiogram, we're really looking at these P waves and how they relate to the QRS complexes to see whether your atria are contracting coordinately, which would come from a normal conduction system pathway, or again, fibrillating like a bag of worms, which we see with these chaotic impulses coming out from all sides of the atria. Got it.
Thank you very much. We have lots of other questions. We're going to try to circle back to some of those as we move on, but we're going to move on now to Dr. Alex Colas. Alex is an assistant professor at Sanford Burnham Prebys in our Development, Aging, and Regeneration program. Much of his research is designed to enhance our ability to take research from the bench to the bedside. Dr. Colas's work is helping to model cardiac diseases in a dish, as well as to devise innovative cardiac regenerative strategies. His lab is also dedicated to identifying and developing new drugs promoting cardiac regeneration and helping to preserve heart function after injury. Ladies and gentlemen, Dr. Alex Colas.
All right. Hi, everyone. What I'm going to talk about is the current approach that my lab is taking in order to try to understand a little bit better what are the molecular causes of atrial fibrillation in human cells. And what we do is to use, as you can see, let's see if I can show you my cursor here. We use human-induced pluripotent stem cells to try to understand how human heart rhythm biology works with specific application to atrial fibrillation. Let's see. So as Dr. Muse was alluding to, atrial fibrillation is the most common form of cardiac arrhythmia in human. He went through the data. I won't go too much into the details. The most important thing with AFib is that there's a number of risks that are associated with with this disease, which include stroke, which is a very serious, and also ventricular fibrillation. The problem right now is that although we are able to when we look at the heart rhythm, to see that a heart is undergoing an event of atrial fibrillation, it is not very well known what genetic defects can lead to the development of this disease in the general population, knowing that, as Dr. Muse was saying, that about a third of us, a quarter to a third of us, will experience AFib in our lives. So, and as a consequence of that, since we don't know really which genes to target, there are no very good drugs to treat atrial fibrillation. And as Dr. Muse was talking about, was that the best standard of care right now are these ablations, which are invasive, costly, and not curative at the end. Therefore, AFib is currently an unmet medical need. So that was the starting point of our reasoning in the lab. And try to understand a little bit the manifestation of atrial fibrillation at both the organ and cellular level, as Dr. Muse was alluding to in the context of atrial fibrillation. You see in these upper chambers, the atrial part of the heart, you see these abnormal conduction events that are leading to irregular contractions. But at the cellular level, what do we know about the molecular or the cellular manifestation of atrial fibrillation? What we know is that there is often observed with atrial fibrillation a shortening of the action potential duration. For those that don't know what action potential duration is action potential is the electrical impulse that will initiate the heart contraction. So if this is too short, then this can lead to the establishment of these rotor circuits that might get the as you can see here in this movie, get the electrical impulse stuck at the same place. So instead of propagating towards the ventricles, you see the rotors that get stuck into the atria and cause the arrhythmia. So, and however, the molecular mechanism causing this shortening of the action potential is not very well understood. And one of the reasons for that is that the available model system to try to understand what can cause this action potential duration shortening are not well suited for large-scale studies to explore these mechanisms. And so, without getting into the details, those models that could be fast don't have really the good cardiac relevance, so we don't know what really the cardiac relevance for that. And those that have those model systems that have good cardiac relevance are very slow and very tedious to work with. So in both cases, we were kind of stuck with an experimental setups that were not well designed to explore the biology of rhythm in human cells. So that was our starting point, and we in the lab and in a collaboration with Dr. Muse and Dr. Larson, I've come up with a multi-model system approach to try to understand a bit better this mechanism that can cause atrial fibrillation at the cellular level. All right, so I'm going to present our attempt to explore and to develop a model that will allow us to study how action potential duration and rhythm works in human cardiomyocytes. So you might know that now every one of us, we can, the cellular biology has advanced so fast, and this is actually, this has given a Nobel Prize to Dr. Yamanaka. You can take any cell of our body and transform it using these Yamanaka factors into a pluripotent stem cells. So these cells are able to differentiate in any cell type. And my lab has, during the past 10 years, has tried to push these stem cells, these undifferentiated cells, to differentiate into atrial-like cardiomyocytes. But it took us quite a while to figure this out, but we have now been able to very efficiently, almost 100% differentiate these stem cells and make them differentiate into these atrial-like cells. In the meantime, what we've developed is using some machines and automation, we have developed a number of model systems that allows to study how the voltage and the electrical conduction works in these cells that we differentiate. So we had to develop some machines, some quantification of some of the parameters that we're interested in. In this case, it's voltage, and be able to basically make this process smooth and automated and fast enough so that we can explore these processes at high speed. So now this system that is described here is improving the speed of discovery and genetic manipulation up to 200-fold as compared to the previous methods that were described before. So we've published a few of these advances in different papers. So now the question is, now we have the cells, some cells that are relevant to study atrial biology. We have some machines and some methods to study voltage in these cells. Can we use this system to study arrhythmia in a dish? Right, so that's what I'm going to show you now, and I'm very excited about it. So this is an example of, of atrial cells that are differentiated in a dish, and you can see that now we've added a voltage probe on these cells, which basically flash with the variation of the voltage, and these different in light intensity can be measured and quantified and are directly proportional to the voltage variation in these cells. So we can therefore measure action potential duration in these cells, which, if you remember, are shortened in the context of atrial fibrillation. We can actually, with this method, follow every cell in the field of view and detect how the voltage variations evolves over time. So we have extreme resolution over the phenomena we're trying to get at. And so you can see now that for each cell, we can generate a movie and study their physiological parameters. So this is now all automated. So although there is not a lot known about the genetic causes of atrial fibrillation, there are a number of families that present some rare mutations that cause atrial fibrillation. And we've used some of this knowledge to try to develop models to see if we can replicate the findings that are found, the manifestation in patients in our system. And what we did was to take this mutation found in a family that has this point mutation, which you can see this is in a gene called KCNA5, which is a channel that conducts potassium ions. And in this context, you see this red bar is the mutation, and what it causes a truncation of the mutation. It does not produce the full length protein. So the patients are left with half of the protein, which is dysfunctional. You can see in their pedigree that all the relatives that carry this mutation have these black patterns, which means that they are experiencing some form of arrhythmia, suggesting that this mutation is a very strong determinant of atrial fibrillation in these people. So what we did was to take some cells from humans, just as I showed you just before, and we've introduced this mutation in these cells. And then we've recorded how the cells behave in this context. So these are the control cells, the normal cells. You can see that they beat regularly normally. And now the cells that carry the mutation, you can see now that they have a lot of different arrhythmia pattern. You can see that there's a beat to beat variation, so short, short, long, or even some cells experience double beats. So a very profound phenotype of arrhythmia in these cells. So now what we did was to take these cells and ask whether we can use this system to try to correct these defects in the cell. And this is, in essence, the general effort that we will push very strong on during for the next few years, because this is our platform to discover new drugs that might correct the arrhythmia phenotypes. And this is a proof of concept that we can do that in our system. So what we did was to test the role of 400 molecules that have been found that are currently prescribed to patients for different applications. And we asked just broadly, can we use these and see if we can find some new antiarrhythmic drug in this system? And you can see that we were able to find some drug that are making the cells less arrhythmic. And this is an example. So you see that in this system, about 30% of the cells were undergoing some very strong arrhythmia profile. And upon treatment, this number of cells was decreased by more than fourfold. So this is an example of the arrhythmia that is seen in these cells. And if you treat them with this compound, now you can see that you are able to revert this behavior towards a more normal beating pattern. So this is an example of the strategy that is currently conducted in my lab to try to understand the molecular causes of arrhythmia in human cells and try to find small molecules that will correct this phenotype. Obviously, this is all done in cells, and this does not have the complexity of a whole organ. And therefore, this is the rationale for us to collaborate with laboratories that have model systems that study these processes at the whole organ level. And Dr. Larson, which is going to speak after me, is focused on taking this knowledge and evaluate how this works in the mouse. But he'll talk about that. So for me, that's going to be it for me. So just in summary, we have established a new platform that allows to study arrhythmia in human cells. We've dramatically improved the speed of this process. We can study the parameters that matter for atrial fibrillation. As I could show you, we can reproduce aspects of arrhythmias associated with atrial fibrillation, and we can also discover small molecules that can correct this phenomena. So of course, this is just the beginning, and we are hoping to expand these operations to be able to improve our ability to discover new drugs to treat this disease. So this is the team so this is my team. You see these two people. So Anais and Michael have been working very hard on developing this system the past 5 years. It's been a lot of work. Chris and Evan, so Dr. Larson and Dr. Muse are collaborators with whom we meet very often to discuss different ideas, different strategies that we might want to take. And this work has been funded by Sanford Burnham, of course, but also by NIH and the CERN. So with that, I be happy to take any questions. Thank you very much.
Thank you, Alex. One of the questions I have is that, so part of what you're doing is you've created now sort of using the pluripotent stem cells, you're creating as close to a human model as we've ever had of studying this disease. Is that correct?
That is correct, yes. This is really a very exciting new opportunity that for the first time in biology, that we have to really try to understand the molecular basis of rhythm, human rhythm, and by putting these cells in a dish that allows us to really explore the genetic component of that control rhythm at the cellular level. This is a level of resolution that we have never had. It is a revolution for our field, and we are very excited to have been able to develop this at the Sanford Burnham, especially since Sanford Burnham has this wonderful screening center that helps us a lot to do the screening that I was talking about.
So if we can generate these cells to do testing on, could we generate cells to fix the damaged cells in our hearts when something like this happens? So
that is currently being conducted, but in this case, we use these cells mostly to model, to try to decipher the molecular causes of arrhythmia in humans. It is obviously very difficult. We cannot do that in human, and even in animal models that approximate human physiology. It is a much slower process to try to explore these mechanisms. So really what these cells provide is a unique opportunity to test how every gene in our human genome might control rhythm alone or in combination with other genes. So right now, the strategy is to use these cells outside of our body to teach us what controls our heart rate.
So your work, you're sort of developing the user's manual for the heart, which then once we know sort of what genes control it, whether it's inherited or not, or could be inherited or not, all of those things can then go back into the lab, either here at Sanford Burnham Prebys or in other places, for people to find solutions to it.
Exactly. So it can come in two flavors. You could, for example, take advantage of the genetic information of a patient. And if we know that this mutation or the dysfunction in a gene that we know that can cause arrhythmia, we can try to model this and apply different treatments and see if that would revert the arrhythmia observed in the patient. We could also take cells from the patient and test them directly in our system and apply a number of different available drugs or new drugs that we are currently developing to see if that could help this patient very well. So basically, so as Dr. Muse was saying, that it is very difficult because we are all different. It is not known if one treatment will work for me or for you. So this ability to be able to test these molecules in a personalized manner is definitely an option that we can provide at this point. This is very exciting, very beginning, very exciting.
And one last question from our audience before we move on, though there are lots of specific questions that we'll get to afterwards, but also the small molecules that we're testing, are they being used to treat other conditions in humans already? Or are they already proven safe? Where are those coming from?
Yeah, excellent question. So we have both scenarios. We can take drugs that have been already approved and try to repurpose them for a new application. That is obviously the shorter path. And then in the case that, if that's not sufficient, we also have the possibility to identify and develop completely new drugs with this system. Of course, this is a longer process, very costly as well, but we can do this, yes.
Thank you, Alex, very much. I appreciate it, and be excited to bring you back for the Q&A with everybody afterwards. Right now, we're going to move on, though, and I'd like to introduce Dr. Chris Larson. Dr. Larson is an associate investigator and adjunct associate professor in our development, aging, and regeneration program. Chris also had the very best response when I asked him to help me describe what he does to a layperson. He said, "I take results from the laboratory and convert them into drugs for atrial fibrillation and heart failure." I can't put it any more simply than that. Dr. Larson, the floor is yours.
All right, thank you, Joe. As Joe said, my research focuses on really two things. One, what goes wrong inside heart cells that causes diseases both like atrial fibrillation, our topic for today, but also like heart failure, another long-standing research interest of mine. And then second focus, how to discover drugs that can help fix those problems. So I'll just do very briefly a reminder of things that both Alex and Evan have told you. As Evan noted, normally the heart beats evenly, so an even interval between each beat at whatever speed at which it is pumping. And this even beating is caused by sort of a smooth and even conduction of electrical signals from the sinoatrial node at the top of the heart, and then down across the entire organ to the tissue at the bottom. And as Evan noted, in atrial fibrillation, the heart tissue is altered, and that altered heart tissue then interrupts and causes uneven flow of those electrical signals, which then cause an uneven contraction of the chambers and a reduced pumping or output of blood. And I like to usually explain this to folks who are outside of the field of cardiology by using the analogy of cooking and a turkey baster that I show on the bottom right. And we've all tried to do this. I love to cook, and certainly one can have a very frustrating attempt at trying to suck liquid up and then eject it out of the turkey baster. But if you have a large volume of liquid, you can very regularly and evenly pull liquid into the bulb, squeeze it out of the bulb, et cetera. That's how your heart would normally pump. And effectively, atrial fibrillation causes that expansion and contraction of the bulb to be incomplete and irregular. So Alex showed you just before me how he uses stem cells to make cardiomyocytes that we can then study in a high throughput manner to make discoveries much faster than we were ever able to in the past. I'm going to go one step back and talk about a little bit of what Evan showed you, which is the whole heart. And I want to just show a cartoon of an electrocardiogram, as Evan showed. I'm not going to go into the details of how these measurements work or why the trace looks like this. I just want you to kind of remember this analogy. Think of the heart muscle cells like a battery. They have a positive and negative charge, and those positive and negative charges are separated, just like in a battery. In the heart muscle cell, those positive and negative charge are separated by being on either side of a barrier. And that barrier is the membrane that separates the inside and the outside of the cell. When those positive and negative charges do what we call a depolarization, which means that separation collapses together, that happens because ions that are either positively or negatively charged cross that membrane and get next to each other and eliminate the charge separation. That's what we call a depolarization. When that depolarization happens, the overall cell itself responds to that loss of charge separation by contracting. And then as that wave of depolarization travels from cell to cell across the heart, just like a wave that you would see on the ocean, that depolarization wave actually causes an electrical current that spreads out into the body. And the electrocardiogram detects that current by having electrical leads placed at various locations in the body that can detect that current. And so this cartoon here just shows you the output of that in a stylized manner. And I just want to show you kind of what each element represents so that when we show you some data, you'll actually be able to follow along. So most important for our talk today is the very first thing that you would see in a heartbeat, which is what's called the P wave. And this P wave represents when the cells in the atrium, so the upper chamber that Alex and Evan told you about, depolarize. So that charge separation collapses or comes together in response to the signal from the sinoatrial node. And you see a little bump here in the trace. That represents the atrium depolarizing and then a little bit later contracting. Next comes the QRS complex, which is when the big chambers of the heart, the ventricles, then that wave of depolarization gets to them physically from the top to the middle, and they now depolarize. And that is going to be the big contraction of that turkey baster to eject blood, the main pumping action of the heart. And at the very end, you're going to see something called a T wave, which is now when those charges separate back apart. So various proteins in the cells pump those charges apart from each other. We use energy. That's one of the biggest sources of energy consumption in our body is the heart pumping those ions back apart to recreate that separation, that polarization. And we see that as a T wave. So now Evan does this all the time with his patients in the hospital. And we can do the same thing in the lab with basically any animal that has a heart like ours. And here I show you the setup in my lab where we do this sort of thing routinely. On the left-hand side, you can see these chambers here. And these are basically a transparent little shoebox with gas coming into them. And this is where we can put mice to sleep for a few minutes so we can handle them. On the right-hand side here, you can see a little white platform with some wires underneath and some metal strips here on top. If you were to go to a cardiologist, you wouldn't see any of this. They would put little stickies on your chest and on your arms, and then they would attach electrical leads to that. And they would then read an EKG off those leads. We could do that with our laboratory species, but actually it's a little bit hard to do without causing damage because they're much smaller than us. So we actually made something different. So here you're looking now down at the top of that white platform. And I thought this might be amusing to some people. So this is actually a cutting board from Crate and Barrel. And we drilled 4 holes here in this cutting board. And then we bought these little metal electrical strips which conduct electricity. And we kind of ran them down through these holes and to attach the EKG clips to the other side. And then we can basically tape the paws of the now asleep mice to each of those little pieces of metal. We keep their nose in this little hose here to keep them asleep, just like if you were put under at the dentist for a procedure. And now we can go ahead and do an EKG without the animal shifting around and causing misreadings. So here now I'm just going to show you a couple of data slides. So that engineering that setup was one important issue to help us support the work and further the work that Evan and Alex talked about. Another aspect of that, of course, is what are the animals that we use to study this on? What's the appropriate experimental system? And so we use, like many people, laboratory mice, and they're popular for a lot of reasons in basic research. I mean, they're mammals, so they mostly replicate human anatomy and physiology with obviously some key differences. They're very small, very cheap, so we can do that on an amount of funding and do them in large enough numbers that makes it possible for us to get good, robust experimental size of study. And most importantly for our purposes today, they also, for a variety of reasons, end up being the species of mammal for which the scientific community as a whole has a lot of research tools to make genetic changes to them. So we can alter their genome, make them into different versions or models or replicas of things that happen in humans, and then study the effects of those genes or of drugs on the background of those genes without either having to use much larger, more expensive, difficult to work with mammals or very risky studies in humans until we have much better certainty that what we're doing is going to be potentially effective and certainly hopefully safe. So I'm not going to go into details about this again, but I do want to show a couple things here. On the top, I'm just showing you now, this is one of those mice that I showed you before. And we see a couple, I only show a couple of the traces that you would normally see off of a human. But let's just focus on the red line here. You can see very nicely that little bloop of the P wave here, and then the big QRS complex. So we see a very normal, even heart electrical activity as shown here. And I've pointed out the P wave of the atrial depolarization. Now on the bottom here, I'm showing you the previous generation, what was the best thing in the literature out there in the world for looking at atrial fibrillation. And that, unfortunately, that model required change in the whole heart. And so while we could certainly see a loss of good atrial activity as shown here, anybody can look at this and see that these two red and blue lines look nothing like these normal even red and blue lines. These animals also have a lot of ventricular issues because the gene change is also in the rest of the heart. So we can't get a good clean look at how any gene that Evan or Alex identifies has in terms of atrial function. So that was one important thing that we needed to work on. How can we improve that? So in addition to the technology setup I showed you before, we also developed a new animal model, a new transgenic mouse model, or so-called genetically modified model, where we use something called CRISPR, which some of you may have heard of as a way to alter the genome of cells or entire animals, to now only change a gene, either modify it or mutate it or delete it, in only those two upper chambers, the atria of the heart. And now we could hopefully ask the question specifically, what is the effect only on atrial function? And ask the question, what does it do for fibrillation of the heart? So I show you here the prior best model, and you can see this is what we call a survival curve. So this just says how many animals survive over how many days after we delete a candidate AFib gene from the whole heart. And you can see here that the majority of these animals don't even live 2 months. So this is a substantial defect. This is, you probably wouldn't know this if you weren't a scientist, but normally mice don't really ever have heart problems. So this shows you this is a significant problem. And on the right-hand side, now I show you just the results of the animals that we made. We're now using CRISPR. We've only taken the gene out of the atria. And you can see that we have no animals dying. So that's good. So we know that this is robust enough for us to study over time. And now, just as importantly, you can see here, again, now looking at a wild type animal, again, pretty normal curve, pretty normal trace, pretty normal red and blue. We also see that nice P wave there. But now down here, you can see pretty cleanly that in fact, this blue line looks pretty much like that blue line. This red line looks pretty much like that red line, except that this little bump of a P wave before the QRS complex is now nicely deleted out. And so that tells us that these animals survive and that they cleanly have this gene missing from their atria, and that missing gene results in exactly what you would predict is a missing electrical response of the atrium. So that's good news, because that sets us up now to sort of, I'm just going to finish up here in a couple minutes to pull back and talk about the larger picture of our collection of labs here again. So now we know that we have all three efforts that we would need to really advance and find new causes, new drugs for atrial fibrillation. We have the clinical expertise and patient samples and knowledge from Evan. We have the basic research and stem cell biology expertise and basic mechanism expertise through Alex. And then I can bring both the animal models and mouse models to really test those genes, and also the ability for us to start out drug discovery. Didn't mention my prior job before SBP was running the cardiovascular drug discovery unit for a large pharmaceutical company. So we're pretty excited now to have all these research tools and individuals together, and that allows us really now to focus as a network, multi-institutional network, on goals that we think are really critical for the field. So discovering new targets for atrial fibrillation, be able now to attach each of these genes to subsets of the disease that folks like Evan see, to now get to what I just mentioned at the end there, which is to discover candidate drugs for as many of these as we can, and then either ourselves as far as we can, or more likely with corporate partners down the road, be able to advance these into clinical trials and onto the market. So someone like Evan, again, could then write a prescription and give it to one of his patients. And we feel pretty good about the start we've made. We've been able to confirm in our system most of the things that were already out there in the literature. More importantly, we've been able to find new genes, Alex and Evan, new genes that show this same phenotype of a disrupted atrial cell or organ function, and now have a good model that will allow us in a high throughput manner to quickly ask what effect does that have on the atrial electrical activity, and then of course jump forward into the early drug discovery that we would want to do. And just my last slide would just be to say that of course we are on sort of this side, drug discovery, and is a long process over time involving many different types of folks. We're kind of over here using, as Joe said, grants and philanthropy to do basic research and early translational research. And in the future, we hope to be able to hand this off or partner up with people who can really bring the expertise and capability and resources to do the big, expensive clinical trial work and also get drugs on the market. So with that, I'll simply end as Alex did by noting the both present and former members of my lab who've contributed heavily to this work, and note that this work has been supported both by grants from NIH and also from money from the Institute itself. So with that, I'm happy to stop and take any questions.
Chris, we've got a better model here now that we can hopefully more rapidly move drugs along. One of the questions here is how long until this San Diego Heart Network produces treatments for the market?
Yeah, that's a great question. So we're already, as you know, we're kind of already underway, and the way this, as Alex mentioned before, we're at the stage right now where we are identifying new genes and the products of those genes, verifying in the cells and in the animals that they alter atrial cell or organ function. And then once those are verified, we can then, as he mentioned, collaborate with the screening center at SPP to run, hundreds of thousands, so up to 1 million compounds in plates, tiny little test tubes, and look for molecules that interact with those proteins. Once we find those molecules, we then take them back into my system or Alex's system and test and see, do they alter the function of those proteins in ways that would be good for someone like Evan would want to see. And then we kind of iterate that cycle. We make small changes, see if that makes them better or worse, and kind of go like that. So I would say we're already underway, but it's certainly at the beginning.
There's another question here about, so where we are now, what's the approximate time horizon? Is this a 10-year process? Is it a 3-year process?
Right. Typically, I'll just speak about averages, and I would speak about this coming from my prior phase of my career. Typically, from the point of the beginning of a project to a drug being approved by the FDA, such that someone like Evan could write a script for it, that's usually 8 to 10 years.
Okay. We're going to move on. We have lots of questions, but I want to get to Stacy. So Chris, thank you very much. And I would like to introduce our very special guest. Stacy Weaver is the Executive Director for the San Diego American Heart Association, working to improve the cardiovascular health of all Americans. Stacy knows the work that the San Diego American Heart Association does every day affects the lives of community members and families, and she joins us today to talk a little bit about how scientific research impacts members of our community. Stacy, please go ahead.
Wonderful. Thanks, Joe, for that introduction. And I'm just blown away by the panelists and the work that is going on. And as Joe mentioned, I'm here to kind of pull through why is this research so important not just specifically to the AFib research that's happening with our panelists, but research in general. And as community members and as the executive director for the American Heart Association, I think about the importance of the outcomes on our life from research that's done just like this, and the collaboration between organizations from our panelists, and what can we expect in years to come. And so the first thing I think is our lifespan and our healthy lifespan. And according to the CDC, we know that the average American will live to be 78 years of age. But it's not just about living to 78, it's really about your healthy lifespan. And right now, studies show that our healthy lifespan is only 66 here in America. And so really what work that is being done, specifically in the AFib research, but also just in research in general, is really bridging that gap between the 66 years of age and the 78, knowing that we all want to have a healthy retirement, healthy life between those ages. So I think about, that's really the importance of this research and the work as we extend that research through to those clinical studies and eventually to the medication that we can take to help us get diagnostics done, and then medication that helps us solve for this. I think the second area, as I think about the panelists and the discussions today, is the work that is being done and how we pull that research through. Not always does research equate to medication or a technology that is built for us in the community, but it's also advises us on guidelines in how we're treated as patients in the hospital. For the American Heart Association, we actually have a program called the Get With the Guidelines that advises medical institutes and hospital associations and hospital systems on how we bring a patient into the hospital and then all the way through to discharge and following that patient afterwards. And so the work that these gentlemen are doing is also going to help us advise that guideline. And again, what questions do we ask when somebody comes into the hospital to do early diagnostics? How can we quickly diagnose somebody with AFib? And then what is the work that has to be done on the external? And I love the story that I heard when I first came in with the American Heart Association. You think back a couple decades ago, if you had actually had a heart attack, you would have gone into the hospital and likely been discharged with an aspirin and a follow-up for your primary care physician. And today, because of research that's done, just like the panelists are doing, we actually have guidelines that help with the discharge information and help those patients get more information on what do they look for at post-surgery, what do they look for post-diagnostics, and understanding how they should take care of themselves in and outside of the hospital. And then I think about the third piece, just the importance of the philanthropy work that we do as citizens here in San Diego and across our nation, and making sure that we think about the research that is being done and extending the work gentlemen are doing to ensure that we really work in the community. And one of my favorite stories, again, is as we think about the grants that it takes and the funding that it takes to do this research. A lot of us automatically think it's millions of dollars. And I think, Joe, you said we wouldn't be opposed to taking $1 million, but it really is small little dollars that it takes to do this research. I don't think anyone would be surprised that some of us have heard that over half a century ago, the first cardiac pacemaker implementation began, and it started with only $2,000 budget. And I think we're all kind of surprised by that it took such a mere amount of dollars to start something that now today 3 million people worldwide are affected with, and over 600,000 pacemakers are implanted every single year. So I think about those three pieces. Is it about the lifespan? It's about how we advise the medical institutes and medical practitioners in our community to help us understand more. And then the third piece is that the philanthropic side of this is that it doesn't take a lot of dollars to make something like the gentlemen, Chris and Evan and Alex, they're doing really come to life. So I'm hoping that everyone on the call can think about that kind of, those kind of effects on your life and on what is the give back that we can do to help these gentlemen get this all the way through to clinical trials.
So we're going to bring everybody back so that we can have some questions. But Stacy, let me start with you. So when you hear about the research, the new models, that Alex and Chris are talking about, how does that, as someone who's seen the whole landscape of research into this area, what is your reaction to some of the work that they're doing?
It's absolutely critical, Joe. The early research and the life cycle of research, it's important for us to understand that, first of all, the collaboration, very often we think about scientific research being done in, just one research area. And the fact that Chris and Alex and Evan are really combining those efforts and consistently having conversations on the back end, it's extremely critical. I think the second piece is just understanding that research isn't done with just one piece and never stops. The research continues and the life cycle of research continues. So the American Heart Association will be part of this in time. There's other research being done, but all of this research is then put together to help us, again, think about the outcomes of a patient and as a citizen.
Super, thank you. Evan, I want to go back to you. We had a couple questions about ablation. One was, what are the issues with ablation that make drugs a preferable treatment? And one was how the ablation procedures work relative to the cell issues described. Can you take a minute or two just to talk about that?
Yeah, so an ablation procedure is going in, is an invasive way to address the atrial fibrillation, and it's where, an electrophysiologist is able to feed catheters up into the heart to try to search for the areas where these arrhythmias are coming from and try to electrically isolate those cells from the rest of the cells of the atrium, okay? So if we can, wall off those atrial fibrillation circuits, then hopefully that the normal sinus rhythm can take over before. We always like to have a discussion about who's best fit for these sort of procedures, right? That would be determined by looking at an echocardiogram to know what the structure of the heart is, right? It also helps to understand, are you having any symptoms and what have you tried in the past, right? It's definitely not my practice to jump into a ablative procedure as the first step, right? It comes through discussion. We want to amplify benefit while minimizing risk. That's not only with medications, but also with any invasive procedures that come with risk as well. So I think it's really a fascinating area of research, trying to come up with what are the best mechanisms and the best procedural aspects that can help us achieve long-term outcomes with atrial fibrillation. But it's something that's going to be very specific to an individualized patient to understand whether it works for their disease process alone.
Thank you. Alex or Chris, do the current models that we have help us understand how existing drugs, beta blockers and things like that really work to currently treat it? And have you used any of those current treatments and seen how they work compared to what we're discovering?
Yeah, I can respond for the human part. Yeah, we have tested most of the drugs that are currently prescribed in human, and they work as you could predict. We're able to see the same effect that are seen in human in the whole body. We're able to see exactly the same effects in our cellular system. Now, of course, since we're trying to explore novel molecules, the big goal right now is to develop and uncover novel chemistries that would do better than existing drugs. I'll let Chris respond for.
Yeah, I would agree. I think the current drugs actually are fairly well understood in how they work, and in animals they do as you would both predict and as we normally see in humans. So I think our main focus will be, maybe I think what the questioner is alluding to will be the idea that we can compare. And one of the really nice benefits of having a good animal model is, as Evan mentioned, when you get into, of course, in clinical practice, you want to amplify benefit and minimize risk. In the drug discovery process, you do the same thing, but you do that kind of over time. And so before you start into a clinical trial, you want to have done all the things that you can do to maximize the probability of benefit and minimize the probability of safety issues before you start those human trials. And so the animal models after Alex's cell models are a great way for us to say, okay, before we put any human being at risk with something that's unknown or never been tried before in a person, let's look at it in cells, let's look at it in animals, and at least see that it's hopefully more likely to be effective because it's more effective in that model, or safer or more tolerable because it's more tolerated in that model. And ideally, we would have both before we then someone went ahead to try that experiment in a human, which is what a clinical trial is.
Alex, the research that you're doing on this high throughput screening to find all these small molecule drugs, what's the cost of that kind of research, just so people understand?
So it takes quite a bit of resources. You need to build up the machines, the methods. It's collective effort is a little bit over $2 million so far to be able to be where we're at this point. A little over 2 million. Probably not that to really push this forward as fast as we would like. Yes, that's so it's a lot, but it's not a lot. I think it's it's what it is.
Sure. Things cost.
Yeah.
Chris, what about your work in taking then what Alex discovers and testing that in your models? What are the costs of some of that?
Yeah, I would say Alex's costs to kind of build his new system were probably higher than mine. The running costs of animals, of course, a mouse is more expensive than a cell. And so the going forward costs that we would do would probably be quite a bit higher. So I would say if we were to do what we were doing, over 5 years and would have studied maybe 10 or 15 genes and several drugs, it would be the same thing. It'd probably be single-digit millions to do all that.
Okay, pocket change, right?
Well, in the big scheme of things, yes, but it's very expensive. But I think Stacy makes a great point. The other hand I could say is, for us to do one high throughput screen on, let's say, a gene that the three of us had come up with, right? If we wanted to walk that gene over to the screening center and pay for a high throughput screen on 1 million compounds, we could do that for $100,000, right? So there's also sort of denominations of work. Now we don't tend to think about that because it's a continuous cycle of doing a whole bunch of things. But if we just kind of focused in on one study and said, what is the kind of ticket that you buy to do that? That ticket can be as low as Stacy said, sometimes a couple $1,000. In the case of us screening a new drug target, could be $100,000. So it really varies, and there's different ways to look at it.
Sometimes it's paying a graduate student to sit at a computer and compile data so that we know the idea we have has legs. So, I totally understand that. Let's take one last question for Evan. Evan, are people generally born with AF, or does it develop over time? And if it develops over time, you talked about some of the preventative options to look at. Is that better? I mean, obviously it's better for people to be able to prevent it and never have it than to treat it.
Yeah, so what we're seeing is that there are some people that are born with a certain genetic load, right, that make them more susceptible to atrial fibrillation throughout their life than others, right? I mentioned the study that we did where we were looking at, a collection of variants in your DNA, molecular markers, right, that we could add up and form what's called a polygenic risk score for atrial fibrillation. And our DNA luckily doesn't change from birth to death, right? So, very early on in our lives, we could understand what our genetic load is for different diseases such as atrial fibrillation, coronary disease, diabetes, and cancers, et cetera. But what we always talk about is nature and nurture, right? Then there's the environmental factors that meet the genetic factors. So that means that as we are young and able, hopefully we're living very active lifestyles, eating the right kind of foods, exercising, and maintaining our health, preventing things like high blood pressure, high cholesterol, obesity, diabetes. But understanding where our risks are early on might make us more motivated during those phases that are 30s, 40s, and 50s, where we're spending time working really hard at our jobs, starting families, right? Doing all these other things. And sometimes the healthy lifestyle gets pushed to the outer edges, right? But this is really the time to amplify so we can see those benefits when we're 70, 80, and maybe even 90, right? So if we understood that we're going into this with a high polygenic risk, maybe we would avoid alcohol consumption. Maybe we would be more likely to watch our weight and make sure that we stay within the healthy weight range, or try to exercise more moderately rather than saying, you know what, I have busy weeks and not. No, this is going to be important for that person at the highest genetic risk. Or even paying attention, right, Joe, to our heart rhythm. Oftentimes we see individuals present for the first time to the hospital with the diagnosis of atrial fibrillation once they've had a stroke. So understanding what our genetic risk is can potentially help us understand what the screening needs to be done, right? Do we need to stop, take our pulse, understand if it's regular or irregular, hold on to a smartphone-based electrocardiogram or a smartwatch? So it's now opening up a whole lot of questions that are personalized to an individual rather than just thinking about the whole population. And that's not only for prevention, but also here in what our group is doing now for treatment as well, which is super exciting.
Thank you, Evan. Evan, can you give your phone number one more time if people want to reach out if they have a specific question?
Absolutely. I'm at here at the Scripps Clinic at 858-824-5112. I am lucky to work with some of the best electrophysiologists and team members in cardiology and research. So happy to meet with anyone to discuss their own condition as they feel fit.
Thank you very, very much. And thank you to all of our presenters, and thank you all for joining us. As always, our presentation was recorded, and we will be emailing that to everyone who registered. You can please join us for our upcoming events. And if you would like to learn more about or contribute to any of our research, please contact me via the email or the phone number on your screen. We thank you very much for attending today. Feel free to share this with your friends and colleagues, and thank you for your interest in our science.